Yeah, so this is the first review paper. And again, this is written, it's an academic paper. I don't expect you all to understand everything. But there are parts of this, especially in the introduction and perhaps later on, that after listening to me here, you should be able to understand. And it will give you a sense of, you can follow what's going on. And in this case, it's just talking about that foraging gene that we were talking about that's related to the rover-sitter polymorphism or behavioral differences. And in this, it gives you a better sense, a broader sense of how important that gene is in terms of influencing behavior, not just in fruit flies, Drosophila, but also across other species. And I'm going to talk a little bit about that now. And I've actually taken a couple of figures from this review paper and added them to the PDF. The slide deck, the PDF slide deck that I put on D2L. And there's another one, and we'll talk a little bit more about this, but this is about vasopressin and voles. So we'll talk about that, but it's the same thing. These are, of course, academic papers, and I don't expect you to read them and understand absolutely everything, but it's worthwhile going through them and just, trying to help sort of solidify your understanding of the ideas that I'm trying to bring to you. Okay. Is there any questions? I guess I should ask before we go on. No? Okay, I'm assuming you guys can all hear me. I should have asked before. Okay, so last time we were talking about the rover-sitter, behavior, these different phenotypes. And I'm just scrolling through some of these slides. I'll just show you, just to remind you that we were talking about two different behavioral phenotypes in the larvae of fruit flies, where in the presence of food, some of these larvae will either be sitters or rovers. That is reflected in their patterns of movement, as you can see here, right? And so this is in part driven by the presence of a gene. And this gene, let me just go back here if I can find it. Yeah, so the gene is this one. It's the FOR gene, FOR, it's short for foraging. And this is a specific gene that seems to have, in this case of fruit flies, has two alleles, one that's called rover and one that's called sitter. And, the genetics behind it in terms of, it seems like it follows Mendelian rules and so on. That is that, rovers tend to be heterozygous and homozygous. That is that the rover allele for the foraging gene is dominant over the sitter allele for that gene. So this is just the equivalent to, the simple Mendelian pattern where you have a single locus with like A1 and A2 or big A and little a alleles, right? So it functions the same way. So it's a very simple genetic system that seems to influence the behavior of these animals. Now, what's interesting is that, now let me just go back, sorry. So yeah, here, so part of the issue is that the product of this gene is this PKG, this CGMP dependent protein kinase. This is an enzyme that affects, that's in the brain. That is that it's expressed in the brain and seems to at least partly regulate the behavior of these larvae. You can treat larvae with this protein kinase, this PKG, and you can get sitters to actually behave like rovers because of the presence of this gene product, right? This enzyme. So the question then is this gene present in other species? And if it is present in other species, does it in some way influence or moderate the behavior in similar ways in different species? And this is from that review paper that I had put on D2L for you. And all this is it's an evolutionary tree, if you will. And you can see all the different species that are listed here that have this foraging gene, or what we might call an ortholog, which means basically it's the same gene. I mean, obviously we don't have the same gene as Drosophila because there's quite a bit of time between us and fruit flies in terms of when we branched apart from each other from an evolutionary perspective. But nonetheless, it's the equivalent gene, right, in humans. And you've got the same equivalent gene in a whole suite of other species, many of them insects, at least that's the ones that have been investigated. And they have very similar functions. So here's just an example. So this is the foraging gene structure and products in species with known behavioral effects. And you can see this again, this is from that review paper. It obviously lots of insects, right? So, but also, mouse, human, obviously, fruit fly, but also, a series of bees, and we'll get into that. Ants, which are closely related, which are more closely related to fruit flies than we are, but also, amphibians. So here's a lab model amphibian, the African clawed frog, for which they've also found behavioral effects of this gene. Now, each species, like this is the vagaries of how geneticists do all this work, but they have different names, right? So the gene may have a different name, but it's basically orthologous or similar, analogous, I could say, to this, to the foraging gene that we saw in fruit flies. So here, let's look at this. This is these are honeybees. Honeybees have, they have different, you might call them occupations, right? That is that there are different bees in the colony. These are eusocial insects. I should take a step back and describe how they work. They're eusocial insects. They have a female, which is the queen, which is the one that reproduces. It's the only female that reproduces. And her daughters basically function to keep the hive or the colony functioning. And they have different occupations, so to say, right? They have different roles. So, they have, in this case, we're looking at two that are, a forager type and a nurse type of role. And they are different in a lot of ways. So they're different in their behavior. So the nursing honeybees they tend to stay in the hive. That is that they don't travel. They tend to care for the larvae. So they're the ones that are actually caring and feeding the larvae that live in the little cells that are here in the colony. These nursing bees tend to be young, from 0 to 10 days old. And in contrast, the foraging types are a bit older. Now, these foragers tend to leave the hive, they collect pollen and nectar and return. And so they're going back and forth to food sources and bringing it back to the colony. So here is just a reflection of the different tasks that are being performed relative to the types, the foraging, or I should say the behavioral phenotypes of these honeybees. The nurses tend to stick around. So here's the age of the bees. You can see the younger bees tend to stick around. The older bees tend to become foragers and move and so that's where it is. So their age seems to be an issue here as well. So we'll talk about age in a minute. So Am4 is what we call orthologous to the foraging gene. So this is sort of like this is the equivalent. Like it's, how do I put this? It's on the same position on the genome, which has been given a different name by people who study genetics in honeybees. Okay, so it's obviously it's not exactly the same gene. Like the DNA sequence is going to be very different. It's going to be different between a honeybee and a fruit fly, but it is sitting in the same spot and it's producing a similar gene product. In this case, it's producing, again, this CGMP dependent protein kinase, this PKG. So this is similar to the PKG that's found in those fruit flies, right? In the larvae of the fruit flies. And so, if this gene product, this enzyme, has a similar effect on these honeybees as it does on fruit flies, then we should expect to see that this PKG, we're going to use PKG as the short form, the PKG is going to be higher in the brains of foragers than it is in nursing bees. I should say that this, one thing I haven't done yet is explain, much of this work was performed by a woman named Marla Sokolowski. So Marla Sokolowski is a behavioral geneticist at the University of Toronto, Mississauga, and her lab have been the ones that have discovered this foraging gene in fruit flies. and have extended much of the work into other species, which is what I'm describing to you here. And Marla is the the senior author on that uh, on that review paper that I put on D2L for you. I was fortunate to meet Marla. She came up and gave a talk here once. She was invited by Thomas Merritt from over in biochemistry, or I guess he's in my department now, School of Natural Sciences. But anyway, she's an incredible scientist. And anyway, all to say that that's who is behind much of this. So yeah, so let's look at the PKG levels in the brains of foragers and nursing bees. So here are 3 colonies. So these are our replicates. You can quantify the amount of the Am4 mRNA, which gives you a sense of how much of that PKG is in the brains of these bees. And the nurses here are in the sort of orangey peachy color, and the foragers are in blue. And you can see that across all 3 colonies, the foragers have more of this PKG. They have higher PKG levels than the nurses do. So we can conclude that the levels of PKG in the brains of foragers are higher than in nursing bees, which is what we predicted. But because they have differences in age, that the reason behind this may be simply because of age, right? That simply that younger foragers have lower PKG. Younger bees have lower PKG than older bees. But here, this is not necessarily true. It seems that young foragers have high levels of PKG. Middle-aged undertakers, which are a different group of bees, they tend to exit the hive. They have high levels of PKG, while middle-aged food handlers, which are a different sort of role in the colony, those that they tend to stay in the hive and they do not. So all to say that it's likely that this is not simply an age effect, that this PKG is associated with those roles that require the bee to leave the colony. Okay. And finally, feeding bees a catalyst to increase the amount of PKG activity leads to increased proportion of foragers. So you can manipulate the behavior of bees just in a similar way as to what you could do with the fruit flies by giving them something that increases the activity of PKG. So, there's a gene, this Am4 gene that produces an enzyme, and it's clear that that enzyme is affecting the behavior of these bees. So possible mechanism: changes in Am4 gene expression contribute to change in roles through effects on likelihood to depart the hive. So what does that mean? So, in older bees, we seem to have upregulation of this gene to increase the amount of the enzyme, the PKJ enzyme in the brain. That leads to those bees departing. Phototaxis, what does phototaxis mean? It means that they are attracted to light, right? That they are, and so if that's the case, if this gene, if the gene product, this enzyme affects the phototaxic behavior of the bees, then that means that, if you have more of this PKG, then you could end up being more attracted to light, which means you end up more likely to leave the hive. And that's how this might be manipulated. Okay, this is not the whole story. This behavior is also affected by other genes related to, for example, as I've said here, juvenile hormone, but also social interactions. So some bees, they may be the more social interactions they have, the more likely they may, when the density of the hive is high, then you may be more likely to see bees taking on that foraging role. So social interaction, the environment related to the social interaction, also helps explain variation in that behavior of leaving the nest and returning. So a couple points. Just this Am4 gene, as I said, it's what we call orthologous to the Drosophila foraging gene. There's high sequence similarity. There's a very similar pathway, and as I mentioned, the enzyme is very similar. So it's cGMP dependent protein kinase, this PKG, but it is a different mechanism of action. So just to the point being that in the fruit flies, the mechanism of action seemed to be just the genetic had different alleles, right, associated with the different behaviors, and there was inherited in a Mendelian fashion. But this seems to be, in terms of these in these bees, the mechanism seems to be related to the increase or decrease of the amount of that PKG. So basically through gene upregulation. And there's even more, this is an example of another well, it's an example of this gene affecting other species. So this is the roundworm C. elegans. So this worm is a model organism for a lot of different research related to development and so on. They're translucent, if not transparent, and so you can really do a lot of manipulative experiments with them. But there's a single, so if you look at the receptor protein associated with, this PKG or cGMP, which is associated with this foraging gene, or has a receptor, and that receptor is called NPR1, and a single amino acid difference in that receptor protein for this cGMP leads to changes in social behaviors. So, you can have social foraging or sort of independent or individual foraging in this species, and that is determined, or not determined, I should say, but it's at least influenced by the receptors for this cGMP that's produced by the sort of orthologous foraging gene. So all to say that the gene, the different alleles, the sort of products of that gene, the receptors of those products, all of those things are manipulating, or not manipulating, they're influencing similar behaviors across a whole different suite of species. And in fact, a friend of mine did his PhD with Marlo Sokolowski. And in fact, he's did a lot of work on this rover sitter business. And he a few years ago was talking to another friend of mine who works on migration in birds. And so they were interested to look at, how perhaps the similar gene in birds influences their migratory behavior. And so there's lots of opportunity to try to understand the effects of this gene on behavior. And that's basically what this is doing. So different effects across different species. Okay, so this speaks to something called evolutionary conservation of behavioral mechanisms. So basically what this means is that common behaviors across different animal groups may be conserved from an evolutionary perspective because of because of a shared common ancestor that evolved this relationship between these genes and the behavior, in this case sort of a foraging behavior. Similar pathways in different organisms with different behaviors and life histories, single gene effects and foraging behavior. I mean, this is all I'm doing is reading the slide to you, but basically the point being that this is evidence to suggest that this is a conserved behavioral mechanism, and it's conserved, the genetics behind that behavioral mechanism is conserved across different species, and that, there's obviously variation in how it works, whether it's just simply based on the alleles, whether it's related to gene regulation, whether it's related to variation in the receptors, but all to say that the pathway, the basic elements of it are conserved. Okay, I'm going to give you another example here. This is not related to this foraging gene, and again, just to take a step back. I may not have explained things as well as you may have liked, but that review paper by Marlo Sokolowski, be sure to review it, and it should help explain some of the points that I've been trying to make here. If I can find it, I'll probably try and find a video that may help as well. Okay, So here's another example. This is the prairie vole. So prairie voles are really interesting. So this is Microtus acrogaster. And in contrast to every other vole out there, what are voles? Voles are rodents, okay? So they're related to mice. And, they live, in this case, obviously prairie voles live in prairie habitat. We have redback voles here. It's a fairly large group of rodents. And the prairie vole is unique in a lot of ways, in part because it engages in paternal care. So most voles, in fact, most mammals, but most rodents are polygynous. That is that males mate with multiple females, and females raise their offspring on their own. Prairie vole is monogamous. That is that males and females pair up. They have lifelong pair bonds. The males function as fathers. They help raise the offspring. They defend the female in the nest. And this is in contrast to every other single vole. So, obviously humans are at least socially monogamous. And so that this, understanding the monogamy related in the prairie vole is of interest, just in general interest to people. So trying to understand how and why this behavior, this monogamous behavior has evolved in prairie voles, or not just evolved, but also how does it happen mechanistically, and from a genetic perspective is a big question. So the prairie vole, the male engages in paternal care, right? So when the mom gives birth, he'll clean the offspring, he'll consume some of the the placental membrane and all this, and he licks the offspring to ensure that it's breathing and so on. And there's a lot of intensive paternal care, and that paternal care is completely absent from every other vole. So, and it even engages in nest defense. So here's some cute photos of prairie voles fighting. So the male is not just engaged in paternal care, it's also engaged in nest defense from rivals. This is a more typical vole. This is the mountain vole, Microtus montanus. It is, like every other vole, polygynous. There is absolutely no paternal care, and the males and the females don't live together. They don't cohabit. Here's a question. Do they still have extra pair copulations? But I guess you're referring to the prairie vole. That's a very good question. I don't know the answer to that. It'd be easy to find out if they looked into that, but it would be interesting if they engage in extra pair copulations. If you remind me, I'll try and look that up. It shouldn't be hard to find. Okay, so the question is, let's compare these two species, right? One which is highly monogamous, one which is polygynous with no paternal care. I say paternal meaning no care from the father, whereas, as I mentioned, the prairie vole does. And so here are these differences just outlined. Lifelong pair bonds, paternal care of young, male defense of the female by the prairie vole, and then the mountain vole, polygynous, no paternal care, male does not cohabit with female. What is the mechanistic basis of this difference in sexual and social behavior? So it's related to vasopressin. So it's a hormone. So here we go. So prairie voles, they require repeated mating to form a pair bond. And that repeated mating results in the production and release of vasopressin in males. And that vasopressin binds to these dopamine cells, dopaminergic cells that produce dopamine in the brain. And so that creates this reward cascade, this positive feedback. So the more sex that the males have with the females, the better they feel about being around her. And so you've got this sort of, this mechanism, this hormonal mechanism that creates this positive feedback or reward cascade that facilitates this monogamous behavior. Now, the mountain vole does not have the same level of vasopressin synthesis and release. So the mountain vole, which you recall, does not have paternal care. it's the one where the male just mates with the female and leaves. They have a completely different pattern of vasopressin receptors in their brain, and they don't have the same positive feedback, which means that because they don't have those vasopressin receptors, they don't end up experiencing the same dopamine rush when they copulate with the female. So here we can see the receptor binding in the prairie vole on the left. So you can see the red parts here, and then you see the mountain vole here. Now the receptor parts are in different places too, so that also has something to do with it. So let's do, you can do a series of experiments, and again, there's a review paper, just to reiterate, I put a review paper on D2L that sort of articulates much of this. So, at least the underlying mechanisms and so on. So it's worthwhile sort of reviewing that as well. So experiment 1 here is, you can ask the question, is vasopressin the key to monogamous behavior? And one thing you can do is you can block the vasopressin receptors by using what's called a vasopressin receptor antagonist. So basically that's a molecule that you can put into the brains of these prairie voles that will take up the space of the vasopressin receptor, so that that receptor cannot actually accept vasopressin when it's released in the brain. So that's blocking those receptors. And in fact, when you do that, there is no affiliative behavior with the mate. So that means that the monogamous pair bond that exists in the prairie vole does not occur. So if you block those receptors for vasopressin, that dopamine rush doesn't happen, and these animals then are no longer monogamous. So this, what happens if you transfer the gene that's associated with that receptor, what happens if you transfer it to the mountain vole and then give the mountain vole vasopressin? And so you can induce, the result is that you can induce monogamy in these non-monogamous, these polygynous voles, by giving them vasopressin and then transferring the gene that codes for the receptor for that vasopressin. And so you can manipulate the behavior of these polygynous mountain voles and turn them into monogamous mountain voles by doing this. So that suggests that the gene that's associated with the receptor is important, right? So that gene then influences the behavior of this monogamous behavior. So again, just to reiterate, though, I mean, the gene itself it's not fully responsible for the behavior. That gene associated with the vasopressin receptor is not fully responsible. There's variation in the that's induced through behavior. So in prairie voles, that social sexual activity is required in order to trigger the release of vasopressin, so that it can be lead to the release of the dopamine through the receptor. And there's other genes that may also be contributing to vasopressin synthesis and release time. So the genes associated with the production of vasopressin obviously also have an influence on the behavior. But on top of that, though, the prairie vole, in order to have the behavior happen, actually have to have sexual activity in order to release the vasopressin, and then the vasopressin can occupy those receptors, and then that leads to that dopamine rush. So all to say then, though, these examples have given you a sense of how single genes can have effects on behavior. Certainly not determining behavior necessarily, but certainly can have strong influences on behavior. I'm going to show you, we're pretty close to the end here. So this is related to reward pathways, and it has application to our understanding of addiction. So, the dopamine release that we saw in the monogamous prairie vole, it is that leads to feelings of well-being and pleasure. So that's a reward system that's driven, in the brain. And obviously there are genes associated with dopamine release and the receptors associated with dopamine. This is just a quote. I'll read it to you, but related to rats, So this system of dopamine release and its production of feelings of pleasure, where it was mistakenly discovered by James Olds, and he was studying the rat brain. And what he did was he accidentally placed electrodes in part of the limbic system. So the limbic system is sort of deep in the brain, right? It's sometimes called the reptile part of the brain. But what he did was he placed these electrodes in these areas where, and he could, the way the experiment that he had done, and as you can see the rat here, here's the wire that leads into an implant into his brain. And every time the rat pushes down on this lever, then that leads to a stimulation, electrical stimulation of part of the brain. And this guy, James Olds, was not trying to look at stimulation of the limbic system, but what he found was that it was, whatever area he was hitting was one that released dopamine. In the end, that's what they learned, because these rats were going crazy. So just to read here, Olds found that the rats would press the lever almost nonstop, as many as 5,000 times an hour. Imagine that. Imagine 5,000 times an hour. Like, do the math, like divided by 60, that's how many times a minute? Oh my God, divided by 600, and that's like, roughly like 9-10 times a second. Isn't that right? Is that right? The animals would stimulate themselves to the exclusion of everything else except sleep. They would even endure tremendous pain and hardship for an opportunity to press the lever. Olds had clearly found an area in the limbic system that provided a powerful reward for these animals. So this reward cascade, we can ask questions about it related to humans, right? Because there is evidence, genetic evidence of differences in dopamine receptors in human populations, in people. And so we call this reward, the idea here is it's related to something called reward deficiency syndrome. And there are two alleles associated with this gene that produces these dopamine receptors. So 75% of the population is this A2 allele, or carries this A2 allele, which produces a higher density of dopamine receptors. And then there is a 25%, or a quarter of the population, are carrying this gene, the allele A1, which has a much lower density of these receptors. When you have a low density of these dopamine receptors, that means that you're less likely to get feelings of pleasure from, we might say, like regular or typical activities. That in order to get that dopamine rush, you need to do more extreme things to stimulate more dopamine to be taken up by these dopamine receptors. So the suggestion or the prediction is that those people that have lower dopamine receptors, lower densities of dopamine receptors, may be predisposed to compulsive behaviors, drug abuse, alcohol abuse, things of this nature, risk-taking behaviors. These kinds of things may be predisposed in people who are carrying a gene that gives them a lower density of dopamine receptors. So allele A1 may increase the predisposition to substance abuse because it reduces the natural reward for engaging in so-called average behaviors, right? The people who are carrying this low density set of dopamine receptors with this A1 allele may be less likely to experience sort of the normal reward using normal or average behaviors. So, they need to do more to get a feeling of pleasure that for the rest of people, that is the 75% that are carrying the A2 allele, they get that same sort of dopamine rush, doing something that we might consider normal. So here are some patterns of the proportion of different populations that carry the A1 and A2 allele. So the general population is 25% carry A1, 75% carry the A2. But you can see that in some of these categories, severe alcoholics, cocaine addicts, cocaine and addicts and those that carry other risk factors, even to a degree smokers, they are disproportionately carrying an A1, they're more likely to be carrying an A1 allele. That is, that their dopamine receptors may be less dense than those that do not have these behaviors. That's not to say that it's exclusive, right? Certainly there are people who carry the A2 allele that have some of these addictions, right? So it's not to say that, you know, having a low density of dopamine receptors is automatically going to make you an addict. And it doesn't mean that if you have a normal density of dopamine receptors that you're not going to become an addict. But there is definitely an effect. Okay, so it depends on exposure to addictive substances, the availability of those addictive substances, and even, you know, the role of stress. So there's a whole bunch of other factors that may dictate whether a person becomes addicted to something or not. But it certainly seems that there is a predisposition towards addictive behaviors in those people that are carrying this A1 allele, where their dopamine receptors are of lower density. Okay, so what's interesting is that this human addictive behavior and the pair formation in voles, they have this common underlying mechanism related to the reward cascade and the production of dopamine. And so, you know, in some ways, it's interesting because, I mean, this reflects this kind of evolutionary conservation that I was talking to you about, like similar behavioral mechanisms that occur across different taxonomic groups, right? That sort of carry a similar behavior and a similar, and it's, you know, in this case, of course, you know, this reward cascade that affects the monogamous behavior in voles also affects the addictive, or, you know, these kinds of addictive behaviors in humans. So one could argue perhaps that voles, these prairie voles, you know, they're kind of addicted to monogamy. They're addicted to sex with the same female because of that dopamine release, right? That they get, that reward cascade. All right, and then of course, you know, this, I already reflected this. This is the PKG pathways and how they have a similar underlying basis for foraging and locomotory related behaviors in a variety of different species. You've got the review paper to look at that. So it suggests that, you know, you've seen this kind of figure in, you know, comparative anatomy, perhaps. These are structural, we call structural homologies. That is that you have similar bones that have evolved in different ways across different groups of animals, and maybe behavioral mechanisms are similar. Maybe we have behavioral homologies. That is that you've got the same set of genes and their gene products and receptors, and they are used in different ways, in similar but different ways, I guess, in different taxonomic groups or different organisms, different animals. Okay, so common underlying mechanisms can lead to radically different phenotypic effects when social, physical, and genetic environment changes. I've sort of made that point throughout this. Generates. So anyway, what this conservation aspect means that evolution of complex behaviors does not require that evolution starts from scratch from each species. We've got this underlying genetic architecture that allows for the expression of particular behaviors as a result of this sort of, you know, conserved genetic mechanisms that exist. All right, we'll leave it at that. We've stopped. Okay, yeah, we finished this component. We'll carry on next week. If you have any further questions about what we've talked about today, you know, feel free obviously to email me. I think